Here we go with part 2 of chapter 14, and we left off on what did Mendel's findings mean, and the last part is that what we can conclude is that there's a dominant allele and a recessive allele. And dominant is the functional protein, the one that's going to be made, that will actually be made and function, and that its function will mask the other allele. The other allele is the recessive one, and it makes either a malfunctioning protein or one that actually is not going to be used in the situation that it's supposed to be. In this chart here, you can see some different dominant and recessive characteristics. For example, eye coloring, one that's dominant over the other is brown. Brown is dominant over gray, green, hazel, or blue. Vision, the dominant trait is farsightedness, normal vision, or the recessive would be normal vision versus farsightedness. And you can see it's actually relative to each other, so the alleles are going to depend on what each is. And we'll talk a little bit more about some of the more complexities of the Mendelian genetics. But for more human traits, there's a website here at the bottom that you can take a look at if you have an interest in that. Some more useful genetic vocabulary is making sure that you remember what homozygous and heterozygous means. So homozygous is when you have two identical alleles for a character. So two identical traits are going to be made through these identical alleles for that character. So that would be a person is homozygous then for the gene controlling that character. Heterozygous means you have two different alleles for that gene, two different alleles for the character. So unlike homozygotes, heterozygotes are not true breeding. So when Mendel was back doing his experiments, the true breeding plants, really what he was using without knowing it, is he was using plants that were homozygous for the character that he was studying, whether it was the color or shape of the peas or the color of the flowers or the height of the plant. Each time when he had a true breeding plant, it meant that it was homozygous. So another thing that we should be remembering or reminding you of is the difference between phenotype and genotype. So genotype is what the actual genes are, what the alleles are, and we signify it with letters often. There are some other ways to represent the different alleles, and we'll look at that example too. But then what is actually seen and expressed is what is the phenotype. So again, phenotype is a description of the organism's trait, what is the physical expression, and the genotype is the actual genetic makeup. So what Mendel was doing was making crosses, and you need to just practice and make sure that you can do some simple and maybe even some more complex crosses. So remember that alleles can be represented as letters, and we use typically a capital letter for the dominant trait and the lowercase letter for the recessive trait. So if we use P's for the flower color trait or characteristic, and then we have the purple being the capital P, and then the lowercase p would be for white, the recessive trait. So if we look at this again, looking at Mendel's experiment, we can represent now his true breeding parental generation here, where we have the dominant homozygous purple, and then we have the recessive homozygous for white. And then our F1 generation, our hybrid generation, is going to get one allele from each of the parents. So we have one dominant and one recessive in our genotypes here. And 100% of our offspring in this generation will be the hybrids from true breeding parents. And then when we have self-pollination or crossing of this generation, where all the crosses will be the hybrid or heterozygous, which will result in the F2 generation that has, well, we see here the phenotype that's 3:1, but we need to remember that the genotype is going to look quite different. So here now, the way that we put together the genotype is by using a Punnett square. In the Punnett square, we take one of the sets of alleles of one parent. We typically put the male at the top here. And so one of the alleles in this box, one allele here from this parent. And then the female is usually on this vertical side. And again, we take one allele, the dominant, and then the second allele, the recessive, and set up our Punnett square in that way. And then simply, we just combine the alleles into the boxes here when we cross. So these two would end up here, these two end up here, these two end up here, and these two end up there. And again, you can see our 3:1 ratio of our phenotype, our 3 purple, oh goodness, where did that go? Oh, it really went flying away. There we go. We have our 3 purple for our phenotype versus our 1 white, so our 3:1 ratio. But if we look at the genotype, we actually see something quite different. In this case, we have one homozygous dominant with 2 capital P's, and then we have 2 of our 4 being hybrids or heterozygous again. They have the dominant P and the recessive P, and then again 1 out of 4 will be homozygous for the recessive. So we say we have a ratio of 1 to 2 to 1, or we have 25%, 50%, and 25% in our genotype. So remember, homo means the same and hetero means different. And this is again showing the same difference in ratios between the genotype and the phenotype. But get familiar and make sure you're very comfortable with identifying what these double P's represent, that they are homozygous dominant, for example, or these lowercase ones are homozygous recessive. Make sure you can use the vocabulary here correctly. Okay, and this is really just talking about the same thing, so we'll move on here. But what we really want to look at, and what that last slide was going to lead into, is what is a test cross. So when we have a flower, we don't know if that flower is true breeding, if it's homozygous, when it is showing the dominant trait. So if we have a purple flower, for example, that dominant color or trait is purple. So when we look at that purple flower, we really don't know what its genotype is just by looking at it. We just know that it's showing the phenotype of purple. So the way that we can find out if it's homozygous dominant or heterozygous is to do a test cross. So to determine the genotype, what we would do is breed the mystery individual with a homozygous recessive individual. And then the offspring, if they display any recessive phenotype, then that means that mystery parent had to be heterozygous. So looking at that written out here, you can see that depicted. So here we have our test cross being our homozygous recessive. This is our known genotype because it's the only one that will show the white coloring. But here we have this purple that could either be homozygous dominant or heterozygous. So we do a test cross by crossing the two of them to see what the offspring look like. And if the offspring end up being all purple, well, then that means the only way that that can occur is if this individual is homozygous dominant. And you can see that here by putting the homozygous dominant here and crossing it with our homozygous recessive, we end up with everything being heterozygous and everything being purple. But if that purple flowered parent is heterozygous, it is capital P little p, then we put it in our test cross and we see that by crossing it this way, we still end up with purple flowers because we're going to end up with some heterozygous. But the one allele of the lowercase p here, or the white allele, when it joins with our test cross, which is also recessive, gives us some white flowers again. So the results, we get half the offspring being purple and half being white, which again tells us that the original parent was heterozygous in that case. And this is again showing that same idea, just depicted in a little bit different way. And you can pause this and study this if you're not following, or you just need to think about what it's saying. But you do need to understand how a test cross, or the results of a test cross, what that actually can tell you about the genotype of the parents. So the simple cross that we have is called a monohybrid cross, and that's when we take one characteristic or one character and study it and cross it. So that could, one thing could be flower color, or it can be the color of the seed, and each of those is a monohybrid cross. But more complicated, and what you should be able to look at and calculate, is a dihybrid cross. So Mendel actually did dihybrid crosses where he followed the inheritance of two different characters. So he would look at seed color and seed shape at the same time. And this is called a di, or two, hybrid cross. So this is a picture showing you a dihybrid cross. And the way that we would write this out is we would put one of the characters first and then the other character second. So we put them together. So you can see with this one that Y represents yellow and R represents round, and these are our dominant traits. And so we have a homozygous dominant yellow and a homozygous round. So we end up here with a seed that's yellow and round. And these are true breeding because they're homozygous. And then our homozygous or true breeding recessive individual has the lowercase y, which gives you a green trait, and the lowercase r gives you the wrinkled trait. So we have homozygous green and homozygous wrinkled. So in crossing those, again, what we're going to end up with when we have true breeding parents, our F1 generation is going to be all hybrid. It's going to be all heterozygous, and which will then show all of the dominant traits. And then in self-pollinating, we actually, instead of getting that 3:1 phenotypic ratio, we end up with a different one when we have a dihybrid cross. What we end up with is a 9:3 to 3:1 ratio. And that is just because we're mixing up. We have a lot more varieties of ways to combine the gametes or alleles in our offspring.